Good morning. This is Donna Messner with the Center for Medical Technology Policy. I think we're going to get started now. There'll probably be a few more people joining as we go along, but we've got a pretty robust group now. I want to thank everyone for taking the time to log on to this webinar. And I believe Sean, Sean is in a different location, so we're doing some virtual coordination here. But Sean Tunis, if you'd like to go ahead and kick things off, we'll advance to the first slide. Maybe do we have him unmuted? We're not hearing Sean.
The conference has been unmuted.
Hey, this is Sean Tunis. Can you hear me now, Donna?
Yeah, we can hear you now. Sorry about that, Sean.
Okay, well, I'm just going to have to go and do the introduction again. Anyway, so thanks everyone for joining us. And it's about a minute after 10, and I'm sure we're going to be hearing lots of beeps as people call a minute or two late. Folks should probably be muted by the system, but it's always possible that you may not be, so you may want to put your phones on mute just in case to avoid background noise. So very briefly, again, thanks for joining the webinar. We're going to be talking about some work that we've been doing at the Center for Medical Technology Policy through our program, the Green Park Collaborative, over the last year and a half or so. The focus of this has been a multi-stakeholder discussion around medical policy and coverage guidelines for next-generation sequencing in oncology. The only context I wanted to provide for this is that, as many folks know, the whole field of precision medicine has gotten a lot of attention, particularly in the U.S. as a result of the Obama administration Precision Medicine Initiative. There's been a lot of funding that's gone to the NIH to develop a large cohort of patients with sequencing done, and then some funding to the FDA to further develop the regulatory framework. And the work around reimbursement policy framework for next generation sequencing has been, at least the dialogue is going on a number of places, but we view the Center for Medical Technology Policy as a kind of a good forum to bring together the key stakeholders, including the payers and health system representatives, to try to come up with some consensus around a viable policy framework for next generation sequencing in oncology. And I guess the main point I wanted to emphasize is that we're kind of looking for a framework that balances multiple stakeholder objectives, and particularly one that promotes the generation of good evidence around the clinical utility and value of next-generation technology inform kind of reimbursement and coverage decisions, while also ending up with a framework that promotes kind of doesn't impose unnecessary friction on the uptake of a class of technologies that I think everybody agrees has tremendous potential for patient benefit as well as health efficiency. So advance to the next slide, just briefly. The Center for Medical Technology Policy is an independent 501c3 organization. Our mission is to make healthcare more efficient and affordable by improving the quality, relevance, and efficiency of healthcare research. So we emphasize bringing stakeholders together to develop approaches to generating evidence that inform decisions by payers, clinicians, and patients. And then a major focus, and particularly relevant to the work on NGS, to develop policy approaches for paying for promising new technologies that also promotes sort of efficient evaluation. And then within the Center for Medical Technology Policy, the work on NGS was performed in the context of a program that we call the Green Park Collaborative. And that is provides a neutral forum to support dialogue and convergence towards consensus around methodologic standards for research, where we focus on real-world effectiveness and value. And again, in this case, the focus of the collaborative, as Donna Messner will discuss, is really around a reimbursement policy framework that promotes both evidence generation and rational uptake of this class of technologies. So with that, I'm going to hand it back over to Donna, and we'll re-engage after Donna's presentation. We have our comments from our expert panel.
The conference has been muted.
So thanks very much, Sean, and we'll go to the next slide. And let me just ask everyone who is dialing in to please keep your phone muted. So here's what we will be doing today in my part of the presentation. First, I'll describe the challenge we've been working to address over the past year in relation to next-generation sequencing, or NGS, the approach we've used, and the recommendations we have so far from a multi-stakeholder process of deliberation. This is what we're calling phase one of the work. Then I'll outline key questions we plan to pursue in this upcoming year, which we're now calling phase two of this work. And these are all questions that arose from the first year of effort on this project. And then we'll hear, after my presentation, we'll hear the perspective of panelists representing a cross-section of key stakeholder viewpoints. They'll share their thoughts on the work so far and what they believe are the most important challenges to address in this next year. And then we'll have some time in the last 10 minutes or so for all of you who have logged on to submit questions and hopefully have some interaction around with the panelists on these issues. And so first, some background. Most of you listening will know that when we talk about genetic or genomic assays, we usually talk about three types of evidence supporting their use. First, you have to make sure that your assay consistently identifies the presence or absence of a genomic variant of interest. That's analytic validity. And there has to be the evidence that the variant of interest, or a combination of them, is significantly correlated with a phenotype of interest. That's clinical validity. And then if your assay is shown to have clinical validity, intuitively it might seem that the assay will be clinically useful. But that may not actually be the case, which is why health plans want to see evidence of clinical utility. There's been some debate over how to define clinical utility. I'm not going to go into the specifics of that debate here. For our purposes, the point is that for health plans, clinical utility generally means evidence that using the test in practice can lead to clinically meaningful changes in patient care and especially to improved patient outcomes. And that's been the focus of our work here in this project. So why is that important? Even before the advent of high-throughput genomic sequencing, there was a bottleneck in coverage decision-making for testing. The review cited here found that a large proportion of genetic tests on the market were never covered. And in fact, many of them were never even reviewed for a coverage determination because they lacked clinical utility studies. What are the causes of this failure to achieve coverage? Well, one might be that in some cases, maybe clinical validity didn't translate into clinical utility. Another might be that maybe test developers lacked clarity over the types of evidence needed to demonstrate clinical utility. But I think a more pervasive problem is a lack of incentives to do these types of studies and a lack of infrastructure to make it more feasible to do them. And if the evidence of clinical utility isn't there, the testing is unlikely to be covered. So, as I said, this bottleneck existed before high-throughput sequencing made the situation more complex. Now we've gone from mutation testing in one gene or a few genes at a time, what I think of as sort of the one question, one test, one answer model, to a new model of testing where we can potentially ask many questions and get many answers per test. Some of those answers may not be relevant to the questions asked or may even be misleading, but some of them may be unexpectedly relevant and potentially life-saving. And for sequencing more than a small number of genes, next-generation sequencing is becoming more time-efficient and more cost-effective than older methods. But traditional coverage policies have tended to be designed around the older paradigm of testing, the one question, one test, one answer model. So the question we asked in phase 1 is, how should coverage and clinical utility be construed for next-generation sequencing in oncology? Here's the approach we use to try to answer these questions. Each step of this process involved a multi-stakeholder participation that included people from the diagnostics industry and clinical laboratories, health plans, patient groups, guideline development groups, professional societies, FDA, CMS, NIH, others. We started with a kickoff workshop in July of last year where we began to sketch out the key challenges. And there's a workshop summary posted at our website for anyone who's interested in looking to see what that discussion was. Then we held a series of teleconferences in which we explored specific questions around the evidence needed for clinical utility. But the bigger question was, what if you have a panel? It wasn't enough to talk about utility for individual variants. In terms of clinical utility, the evidence will support the use of some genes in some indications and not support the use of the same genes in other indications, or the evidence will be non-existent either way for some gene indication combinations. So should this characteristic of panels disqualify them entirely from coverage, or is there some kind of workaround that we can come up with? Would payers have to dictate which genes can be sequenced on panels they're willing to cover? Would they dictate which clinically relevant genes could be reported out to clinicians from covered panels? And what about the analytic differences between platforms and between laboratories? Should payers dictate analytic performance standards for coverage? So we were asking these types of questions when we put together a strongman coverage policy framework that we debated in a conference this past April. And then we refined and revised the framework in a multi-stakeholder teleconference, a couple of them held in May and June. The result was the model policy recommendations that I'll now summarize. So, as I noted, payers would like to have confidence in the between laboratory and between platform consistency and quality of NGS testing. But most health plans are not equipped to police analytic performance standards. But part of what we were able to do this past year was provide an opportunity for dialogue between the College of American Pathologists and our Green Park participants. So CAP shared their plans for an accreditation program for NGS testing that is planned to start in 2016. The program includes proficiency testing on both wet bench and dry bench competency, meaning both the assay and the bioinformatics. So we felt that this dialogue contributed to a greater comfort that in the near future legitimate oversight will be in place for analytic performance standards for NGS testing. So the first recommendation is for health plans only to cover testing done by laboratories accredited by CAP for NGS. Now, there still needs to be some discussion on how to create greater transparency for payers to know if some laboratories are repeatedly unsuccessful in proficiency testing. And there's still the question of what additional oversight the FDA will provide in this area. But this seems like a reasonable first step to fill this gap and create more trust in the testing done. The next question, as I noted before, was how we should think about coverage for panels. Generally speaking, the payers in our group did not think it was their role to dictate what panels should look like, nor to limit which genes could be reported back to the clinicians or the patients. They also felt it would be onerous to pick and choose which genes on a panel might be eligible for coverage. In the end, they said the most practicable approach would be to cover panels that meet the criteria for the new American Medical Association CPT codes, which came out earlier this year, for panels composed of 5 to 50 genes. So the Green Park recommendation is that once CAP certification program is in place, payers should begin to cover the 5 to 50 gene panels, so long as at least 5 genes on the panel are guideline directed for the clinical indication, meaning they have demonstrated clinical utility, and the panel cost does not exceed the cost of sequencing those clinically indicated genes by other methods. It's worth noting here that covering panels in this way creates an opportunity for learning from the extra data generated, and I'll talk about that more in just a minute. So what about larger panels? To this point, we were not able to achieve any consensus on covering panels having more than 50 genes, when right now smaller panels can do the job of covering guideline-directed care and oncology. In our discussions, though, some of our stakeholders were convinced that it's better to do comprehensive tumor profiling at first diagnosis rather than waiting until patients have advanced disease and are heavily pretreated. According to these stakeholders, waiting until patients are very ill before looking at the full range of oncogenes and potential targets for therapy really does a patient a disservice. But other stakeholders, including our colleagues from health plans, said that the benefits of this approach still need to be demonstrated. In other words, according to these stakeholders, this is a hypothesis that still needs to be proven. So for the time being, the Green Park recommendations suggest that payers should consider covering larger panels when there are no guideline-directed alternatives, but there is no consensus around this suggestion. So it's a topic for discussion in the next phase of work, phase 2, which I will briefly summarize now. Another issue for phase 2 is how to learn from the extra variant and outcome data that could be gathered from covered panels, and how to promote the development of high-quality genomic databases generally. This kind of learning is critical for more rapid translation of genomics. Some questions to ask here are, how can payers use policy levers to facilitate high-quality data collection? In other words, how can they be partners in this process? And how can the quality, consistency, and interoperability of the data be assured? As I noted before, the appropriate role for larger panels is still an open question, as is whole exome and whole genome sequencing. In this past year, we've been focused on oncology, but for larger scale sequencing and discussions around this topic, we may want to broaden the conversation to include other conditions such as rare diseases. I think that's probably going to be necessary. And it's going to be important to ask, what evidence will support this kind of sequencing? Also, if we put the energy into developing an infrastructure for high-quality registries and observational studies, will this evidence be accepted for coverage decision-making? Finally, some of our stakeholders have suggested we should look at how to pay for off-label use of molecularly targeted therapies when it's clinically justified for patients. Can pharma companies and health plans find common ground in a risk-sharing agreement where pharma provides the drug to patients for the first 3 months and payers begin to cover in the 4th month if the patient experiences benefit? These are topics that we plan to tackle in the upcoming year through the Green Park Collaborative. We're happy and really welcome your thoughts and suggestions. And at the end of the panel discussion, you'll have an opportunity to submit questions through this webinar. And we also want to provide an email address at the end of the webinar for sending us feedback and suggestions after the fact. But first, let's hear from our panelists. So I'll turn it back over to Sean at this point to introduce our panelists. I'd ask again, because we're having some technical issues, we're going to have to unmute everybody on the call to hear our panelists, and that means everybody except the panelists really need to keep their phones muted, please. So we'll pass it back.
The conference has been unmuted.
Hello, this is Sean again. Just checking that you can hear me.
Yep, we can hear you, Sean.
Okay, good, great. Well, thanks, Donna.
Actually, Sean, can you speak up? It's a bit hard to hear you on the other line.
Yeah. Is this better now?
A little louder if you don't mind, Sean.
not.
Okay, let me see.
Oh, getting older and not hearing as well.
Okay, is this working now? Better?
Yes.
Yeah, that's much better. Thank you.
Okay. So great. So thanks, Donna, for that kind of a very quick summary of what was a huge amount of work that was accomplished since last July. The one thing I want to kind of help folks understand is that when we talk about a recommendation of the Green Park Collaborative, what we've tried to do is describe the sort of conclusions that there seem to be a lot of consensus around across all of the stakeholders participating in the collaborative. So it's really a summary of a point of view or a recommendation where there was a lot of consensus. And then Donna mentioned a few other areas where ideas were raised, and that there was some beginning of convergence behind those ideas, such as some of the ideas around coverage of off-label uses, where in phase 1 we didn't get far enough towards actually getting a strong convergence of views. So what I want to do now is introduce our panelists. We have 5 folks who are each have unique and important perspectives related to the uptake and use of next-generation sequencing in oncology. All of them have participated to some degree in the work in phase 1, and each is going to kind of make about 5 to 7 minutes of comments, from their perspective on the work that has been done to date, how it relates to what the work that they have ongoing, and what they think are some of the key issues for the next phase of work that would be could be usefully supported by through the Green Park Collaborative Forum. As these folks are speaking, you may want to jot down jot down questions that you may have at the end. We're going to be giving you an email address to send them to, or you can put them in the chat box, but you might want to be thinking and jotting down the questions that you're going to want to raise for later during the discussion. So first, I'm going to ask Dane Dickson, who's the Chief Executive Officer for MedC, to offer his initial comment.
Great, Sean. Thank you. Can everyone hear me okay?
Yes, you sound good.
Okay, great. First of all, Sean and Donna, thank you for inviting me to be part of this historic web conference. I appreciate the opportunity to discuss what's happening with the Molecular Evidence Development Consortium. The Molecular Evidence Development Consortium is probably one of the greatest initiatives that no one knows about that's currently taking place in the nation and probably anywhere in the world. And basically what the Molecular Evidence Development Consortium, also known as MedC, is it was started as a implementation arm of molecular medicine. In other words, what was happening is MedC could see that there was a great need to bring together all the stakeholders that had a role in implementing personalized medicine. And because there was no existing group that could adequately represent all of these individual groups in an implementation strategy of something such as next-generation sequencing, this 501c3 nonprofit organization was started. In essence, what MedC is, it's a consortium that brings together the 6 major groups that we consider essential for implementing personalized medicine, not only in the oncology arena, but also in any other disease state. That includes pharma, payers, the laboratory industry, test developers, regulators, and then last but not least, of course, there are the patients who should be the end point of anything we're trying to do here. And what MedC has been doing is been providing a framework for which some of the recommendations that were first introduced by the Green Park Collaborative and other groups, it provided a framework for the implementation of those guidelines. And MedC is in the process of building a next-generation sequencing registry that we hope to launch sometime early in 2016 with our first pilot sites. And that registry would allow high-quality next-generation sequencing to be introduced into patients with certain disease states at the time of initial diagnosis. It would be done in labs that have been shown to be able to have a high-quality product that is being delivered. And in essence, what we define that high-quality product is that the lab is getting reasonably equivalent results to another lab that is down the street across the nation or across the world. With that high-quality testing, the laboratory would report that testing into a centralized database, and the clinicians who are ordering the test would also, in exchange for having access to the high-quality testing, would report high-level outcomes such as time to progression per treatment and overall survival in the patients. Then what would happen is this database that would be de-identified of all patient identifying information would be a database that would allow us to deal with the variants of unknown significance, as well as many of the other mutations and alterations that are outside of what the regular providers would consider something that would have clinical utility. Because of the open access, because of the de-identified nature of the database, but yet having genomic data tied to outcomes, this would be a prime database for research opportunities, and the MedC Consortium is committed to making this an open access database for research. As we are building this, we are moving forward very quickly, and one of the things that Sean and Donna brought up is what do we see as the biggest challenges that are facing us as an industry that need to be addressed in the next year. Well, one of the things we've seen over the last several years is we've seen a lot of discussion and a lot of theory surrounding what needs to take place with advanced multiplex testing. It is time that we implement this testing in a way that we can advance science, and so MedC is moving very quickly to implement its database. One of the projects that we are working on right now, which we would ask to be involved with, that we would hope that Green Park would be involved with, is we're putting together a multi-stakeholder group of individuals to define what is the bare minimum clinical data set that is necessary to really make a real-world data set valuable to the individual groups. We have received feedback, and we have received agreements for participation from many of the stakeholders of both regulators, thought leaders, academic, as well as physician groups that are willing to support this. During that work, we will standardize what we believe to be the clinical data elements that are necessary. We will also be discussing what is the format of data that should be put together that would allow further database sharing as we move forward. And with that, I'll end my discussion because I think I've gone over a minute or so.
Great. Well, thanks so much, Dane, and of course we'll we've been in dialogue about the work that you just described, and we think it's a good area of shared interest and shared sort of by bringing together, I think, our various stakeholder networks, we can kind of have the reach necessary to help define those minimum data elements. So look forward to working with you on that. Next comment from Mike Kolodziej from the Director of Oncology Solutions at Aetna. So Mike, are you on?
I am, Sean. Thank you. Everyone can hear me okay?
Yes.
Fine, Mike.
Great. Good morning, and again, thank you to Donna and to Sean for this invitation. As Sean mentioned, I'm the National Medical Director for Oncology Solutions at Aetna. I've been in that role 3 years, and as many of you know, I'm also an oncologist. I always start my talks by saying that the views that I'm about to express are mine and mine alone, do not necessarily review those of Aetna. But that said, my task is to sort of give a more global view of this area as seen through the eyes of the payer. And I will say that Donna has correctly, I think, identified some of the challenges the payers have, and they fall into two major categories. The first is there's a lot of tests. They're really complicated, and we're not smart enough to know whether they're being done well or not. We just don't have the bandwidth we don't have the expertise to look at individual laboratory performance or individual test performance from a purely analytic perspective. And the second is there's a either healthy or unhealthy skepticism about exactly how much the tests bring to the table in terms of improving the health and the care of cancer patients. Now, that may sound heretical, but I would say that those two factors have created what Donna has called the bottleneck from the perspective of the payers in terms of moving forward. I will say that I get invited to a lot of meetings on this subject, and I believe there has been actually tremendous progress made in the last 12 to 18 months in terms of defining a path forward, and I like to recognize the work of the Green Park Collaborative as well as a lot of other groups, including the Tapestry Network and ASCO and many and Dane's group, in terms of understanding how we might move forward. To my mind, the things that have been the greatest successes include, first of all, the recognition that we really, really do need some sort of proficiency standards. It's simply not good enough to buy a sequencing machine and hang a shingle. I think the idea that whether it be the FDA or the professional societies or other accreditation bodies like a new and improved CLIA or New York State or somebody will take ownership of this and will provide authoritative information to the payers to allow determination of entities that should be paid for their work is absolutely a tremendous improvement, and I would say is perhaps the greatest achievement. The second achievement that I want to call out is the recognition that the data set as presently constituted is encouraging and exciting but not definitive in terms of documenting efficacy. What I mean by that is this. We really don't know whether or not you should do a hotspot test or look at the whole genome. We don't know that. I mean, it's clear that when you look at the whole genome, you find more mutations. The question is, does it actually matter for the patient? So that's a really important question. Now, there may be examples where looking at the whole genome does, in fact, make more difference. But I'll tell you, there are more cases where we don't know that as opposed to where we do know that. The second thing is the recognition that just because you have a mutation doesn't mean that it is actionable in the sense that a treatment which targets that mutation yields a superior clinical result for the patient. And I think the recognition that we need to actually learn about that some more is a very healthy academic and ultimately very practical discussion regarding the benefits of our patients. Now, with respect to that second comment, two important solutions have been identified. And those solutions really revolve around trying to identify a methodology for collecting information regarding what type of test is performed and collecting information about what type of treatment is administered and what that outcome for that individual patient has been. So there is now an enthusiastic, multi-pronged attempt to start to collect that data. It has become absolutely crystal clear that it's going to involve a lot of patients. The data is going to be big. Now, why do I say that? Because if you look at the data that has been published thus far, and I will tell you that in my opinion, most of that data is actually both lousy and non-contributive to the moving the science forward. We have very small basket trials that look like they're failures. We have the trial recently published in Lancet Oncology, which is a terrible study. We have the study in New England Journal looking at BRAF, which is interesting but is inadequately powered to answer questions. We have really inadequate information that only tells us one thing. We need a lot. We need a lot of information, and we need to get it all together, and we need to learn from it. So what's the path forward? So we've come this far. We have some general agreement. What's the path forward? And I would propose that there are actually six things that we need to do in the next year 2 of Donna's master plan. The first is we need to finalize the proficiency testing. FDA needs to decide what they're going to do. CAP, I'm very encouraged by CAP's entry into the space. I think we need to finalize that and develop a communication plan. The second thing, which is really, really important, was mentioned by Dane. We need to move forward with this concept of a rapid learning system. We need to make sure that Dane called them core elements. We need to harmonize them. I don't care, actually, whether NCI Match and ASCO's paper and Genie collect different data elements, but they need to collect at least same core elements to allow their data to be pooled and interpreted. Otherwise, actually, we'll have many, many inadequately powered studies, and we actually won't find the truth. Particularly, we won't find the truth about rare events and exceptional responders. The third thing is I would propose that the test developers adopt a policy where they look to get to that magic number of 5 genes. Because I think what Aetna has decided, and I think I'm very proud of this, is if you can tell us you got 5 actionable genes that really are actionable, we're going to cover it for that tumor. And so we cover the 5 to 50 code for non-small cell lung cancer, and we cover it for hematological malignancies. I think you'd be hard-pressed, in the absence of anecdote, to do the same thing for breast cancer. So I think a redirection, thinking about what genes should be included in that 5 to 50 panel with a short-term aspiration of getting it covered by having 5 genes, but also looking at the other 45 genes to look for actually really high-value targets could be a real way to move forward. The fourth thing that I think we need to really think about, and again, this has been mentioned by both of the previous speakers, is the test is actually a tiny fraction of the cost of care. We need to think about how we're going to pay for the therapeutics that are dictated by the test. We need to explore value-based constructs, whether that value-based construct involves pharmaceutical companies providing on a compassionate basis to trial, or whether it involves some sort of value-based reimbursement based on an N of 1 model. I don't know what the right answer is, but I think we need to further that because once we get past the who gets tested hurdle, we will continue to be faced by the who gets the treatment hurdle. The fifth thing is we need to start getting comfortable with regulatory reform, and I'm pretty sure there are people from the regulatory agencies on the phone. When I say regulatory reform, I don't just mean the government. I think the private payers need to start getting comfortable with what constitutes adequate evidence to allow coverage for a test and or a treatment. And I think the old rules regarding randomized clinical trials and what have you probably are going to need to evolve, and they probably need to evolve in the short term rather than the long term. I would call out NCCN for taking a leadership position and start thinking about how they can help us understand this. And finally, I think we need to think about value. So everything we've discussed today is actually based on efficacy. Does it work or does it not work? But that's only step one, and as we think about how this fits into the bigger picture of healthcare reform, we need to understand whether this is better than what we're doing. Is it better than? And I would argue that accepting that it's better than based simply on face value is probably going to be an issue. I think we need to think about how do we look at the results, how do we look at the cost of care, how do we look at the best treatment outcome, best treatment experience for the patient, using novel technologies as opposed to the standard approach. And so with that, Donna and Sean, I'll turn it back to you. Thanks.
Well, Mike, thanks so much. That was kind of a master class in covering the issue. So that was really well done. And I know we and everyone, really appreciates your kind of willingness to engage, put so much of your time into engaging in this dialogue because, obviously the perspective you bring from a large payer is, critical to this. Just a reminder, there definitely a little bit of background noise, a few coughs, a few side conversations. So just double check that everybody's muted who's not talking. And the next comments are from Jeff Allen from the Friends of Cancer Research.
Great. Good morning, everyone. Thanks to Sean and Donna and the team for putting this together and for inviting me to participate in the webinar, and certainly congratulations on the work to date. I know that this was the product of a lot of long discussions, thoughtful thinking from your working group, and really a consensus-driven process to put out the recommendations that you have, and really want to commend that work. It hasn't necessarily been an easy topic, as everyone on the phone knows, but congratulations for really moving the discussion forward. From our perspective and our work in this area, I think generally, we're certainly all familiar with the promise that is accompanying these technologies in terms of advancing personalized medicine, finding new potential druggable targets with the hope of improving long-term outcomes. But I think we also need to recognize, it has been mentioned, that by conducting all of this research in existing silos, we continue to introduce the potential for a fair amount of variability between what we're actually measuring here. Donna mentioned in her opening remarks that the current one test, one answer type approach to companion diagnostics and targeted therapies in oncology is rapidly being potentially replaced by some of these gene panels. But I think it's really important to acknowledge that even in our current system of one test, one answer, there still is a great deal of variability due to the number of tests that are on the market that are claiming to do the same thing, that we really don't know if they are doing the same thing. And this situation is certainly going to be exacerbated by having so many other data elements that are included here. One area that we had established a working group through Friends of Cancer Research that dovetails very well with the GPC was to look a little bit earlier in the process in terms of facilitating the development and use of curated genetic databases. And I think it's important to think about this from a start to finish. So, Sean and I have had discussions along the way about how this work can dovetail where some of the things that Friends of Cancer Research was focusing on was how these data sets can be built to allow them to be potentially used in a regulatory grade, so to speak, for future decision making by the FDA. Certainly an area that the agency has put a great deal of thinking and different proposals out there as well as these data sets are being constructed. And because there are so many different data elements included in this that can range from unknown significance to a research tool and hypothesis generation, all the way toward impacting routine care. But certainly, one thing that I think needs to be considered, particularly for those variants that are being used to guide treatment decisions, is how to introduce systems to measure the potential equivalency between these different testing platforms. We certainly have seen variability between different labs, but at least for those analytes that are being used to guide treatment decisions for targeted therapies, what systems can we put into place to be sure that Hospital A is measuring them the same way that Hospital B is, so as a patient you can feel confident that either institution that you go to, you'll be getting the same results and the treatment decisions made on them will be the same. In terms of next steps, I want to really applaud Dane's efforts with MedC to try and to utilize the work that's been done by the Green Park Collaborative and implement these standards as a way of really trying to get them operationalized. I think it's a very difficult step to move from the theoretical discussion to actual implementation, and I think that the MedC effort is one that is really trying to rapidly test this prototype and try and ensure the consistency and equivalency of different tests to really understand the value of what we're moving forward. And I certainly look forward to working with GPC and commend you for taking on the tough questions that Donna began to outline and how we begin to demonstrate that these tests are providing value. So I'll turn it back over to you, Sean. Thank you.
Great, thanks so much, Jeff. And your kind of all the work that you all have done in this space with the Friends of Cancer Research and your extraordinary ability to bring stakeholders together around, particularly in the issues that are developing large trials with NCI, and the work you're doing with FDA is fantastically important, and it's really coming across to me how I forget who said it. I think it was Mike. How there's many different organizations working in adjacent spaces trying to solve different sections of this puzzle, and I think all of them are necessary to kind of achieve the result we're aiming for, which is the kind of efficient and effective uptake of this kind of class of technology. So it's nice that we're all able to stay in communication and try to find the places where we can both collaborate and coordinate our effort. Next up, we have Vincent Miller from Foundation Medicine. So looking forward to your comments as well.
Thanks, Sean. And if you can't hear me well, Donna, that was never seen such a balanced presentation amidst so many stakeholders. So that was really a great opening act. I thought, the year is brought with it many, I think, observations, initiatives that are shaping the field at a warp speed pace. And some of the articles that have come out recently, I think, are incredibly germane to this group. I was really taken and impressed with the JAMA Oncology editorial from Rick Pazder, Liz Mansfield, and Gideon Blumenthal that talked about the potential of these broad-based approaches that are accompanied by appropriate analytic standards to really drive our understanding of these rare variants and drive patients to therapeutic clinical trials. And one of the things I'd like to see this group tackle is forget about off-label use agents for a moment, because that's not our mission at Foundation Medicine. We'd like to see everybody get on a therapeutic clinical trial. We now have evidence that matching patients to clinical trials molecularly, by and large, not for every given patient, leads to better outcomes than empirically placing individuals on such trials. There's more than close to 1,000 trials of putatively targeted agents across more than 150 genes. And in each of those genes, if you use a broad-based approach, as Dane and others have touched on, you can basically have an infinite number of variants. So how does this collective stakeholders view these therapeutic clinical trials in diseases maybe that have a dismal prognosis, but there are no biomarkers? Say if someone has a metastatic salivary gland tumor, is there not value to this broad-based genomic profiling approach in getting patients onto molecularly matched therapeutic trials, completely understanding that some will not be successful, some will, but that the bar for entry into that space is exceedingly low in some of those diseases. Now, and this leads to this concept, indeed, of a universal companion diagnostic, and which is sort of the other end of what Jeff touched on, of the one marker, one drug approach being obsolete to a large extent. And the idea of a universal companion diagnostic would be to migrate or show equivalence across given CDXs with a broad-based platform, then add in other agents that are other markers that might be tied to therapeutic clinical trials, but also have a subset of genes that have maybe analytic validation established, but clinical utility or relevance, but just pick P53, not established yet. So that's something we're exceedingly interested in and would love to see this group tackle on the clinical trials front. The second point I think I'd make, and something I think Dane addressed it nicely, and it ties to what I said, is the fundamental difference, not only in testing, but in the downstream effects of this comprehensive or broad-based genomic profiling approach from that of a hotspot test. A hotspot test, by definition, looks at predefined alterations that have known associations with a drug, sensitivity or resistance. So even if done perfectly, generates very, very little new knowledge. Now, I know payers generally don't pay for new knowledge. They pay for existent relationships. But I think a lot of folks on this call are clearly vested in defining the power of this new knowledge, because many of these variants will fall by the wayside, but many, many others will have as powerful or more powerful association with given drugs. It's just they're sufficiently rare that no one looks for them, or one doesn't pay attention and collect the outcomes on these individuals. I mentioned one initiative we've undertaken at Foundation Medicine, and that some of you may have seen the press release on it, something called the Precision Medicine Exchange Consortium, which is a group of academic and community sites who've realized that even though we provide great cancer care, we believe we can't figure this all out by ourselves. Simple combinatorics tells us that we need 50,000, 100,000 patients to be able to get a reasonable numerator and denominator for individuals with rare variants. And so a component of this, similar to what Dane was describing, is defining sort of the minimally acceptable data set, the relationships between the individual institutions and foundation and the individual institutions and one another, and having the platforms essentially built that allow this least common denominator to function in a clinically relevant and temporally relevant manner. So I think for me in the coming year, which would be my first year working directly with TPC, it's covered by other foundation members before, would certainly like to see maybe a model developed where payers can adapt it to differentiate what different players in the space are doing, that who captures more of these variants, whose test is applicable to these different settings, and particularly around something like the value, perceived or real value of getting patients unmatched therapies. I think to support evidence generation schema that acknowledging cancer as a disease of the genome and the need for clinical utility to be viewed as something that emanates from the diagnostic pathway solutions rather than the drugs per se, and then to support policy initiatives that reward investment and innovation with a sustainable coding covered and reimbursement pathway. And I thought it was great that Mike called out the difference between someone who just buys a sequencing box and plugs it in. Sequencing is a tiny part. It's necessary, but it's far from sufficient part of what we do at Foundation Medicine every day. So those are some high-level comments. Hopefully there were a few themes there we can capitalize on, and I look forward to fielding any questions.
Great. Well, thanks very much, Vince. And definitely hearing this recurring theme in this space about the critical importance of finding ways to to kind of more efficiently generate evidence, whether by matching patients to trials or kind of ways to link or make standardized anyway these various data collection efforts. But that theme in this space that what we really need to continue to think about is the policy framework that is promotes evidence generation as opposed to a more traditional sort of reimbursement policy framework that assumes the evidence is going to be close to perfect when it comes in the door before decisions are final decisions are made. And that's obviously a challenging paradigm, but from all the comments that have been made so far, it's clear that that's an important part of this puzzle to continue to clarify. So our last panelist is Robert Dumanois or Dumanois. I'm not sure how to pronounce it, but sorry for messing it up. And from ThermoFisher Scientific.
That's just fine, Sean. Appreciate the intro and the opportunity to both continue the applause to the work that Green Park has done, and try and I guess back cleanup within this esteemed panel. We'll keep it moving with a few quick comments here. The first one, in terms of what did the CMTP Green Park crew get right over the last year and a half's work. And I think it's abundantly clear to those who followed, even within this call, there are extraordinarily diverse voices present in the work that's happening here. And when I speak of diverse voices, I mean in terms of panel size, in terms of configuration, in terms of use cases for molecular testing. Tremendous diversity as it pertains to what should be covered, what should be coded, how it should be reimbursed, both in the context of the cost of running one of these tests and industry's perceived belief in terms of what should be there as far as profitability is concerned. Tremendous diversity among evidence that's required, as we've heard, as well as the needs of the ordering physician. What's appropriate for an academic physician may well be very different than those in the community. And we've also heard, even this morning, some comments around this notion of analytic performance associated with testing. And a lot of the, I would say, both opinions and to a lesser extent, some of the poaching that's happened with regard to concerns that exist in the industry. Secondly, I think what Green Park really did get right was the notion of creating a very practical outcome and one of substance. And as evidenced by what Donna shared, it was a lot of noise, a lot of very strong and good opinions, but to end up with a work product that most all of us feel quite good about is great. And lastly, what I talk about here is the creation of good policy, which was not a particularly elegant process, but at the same time, we also saw some of the goals of this project evolve midstream to get to this output, which I think, if you're running for political office, that makes you a flip-flopper these days, I guess. But practically, it resulted in something that looks a good bit better than I thought we'd be, certainly at this time a year ago. And then secondly, in terms of, kind of practically what came out, I think Donna did a nice job articulating, what this coverage policy advocates as far as these panels with somewhere between 5 to 50 markers. And while, to some extent where we are in November feels good about this, the simple reality is 12 months ago, with the exception of maybe one payer in the United States, nobody had a positive coverage decision as it pertains to next-generation sequencing in solid tumor oncology. And the fact that we've articulated some guidelines as far as utility, payment levels, what should be covered, what doesn't have to be addressed in terms of the construct of the test is awfully exciting from my perspective. And a lot of that has evolved really in the last 7 to 8 months. And I think there are other voices who've contributed to this, but the ability to clarify this content and engage a lot of major thought leaders on this front is extremely important. Yet at the same time, stayed away from issues pertaining to whole exome, whole genome, and issues with either off-label therapy prescriptions and clinical trial support. So, kind of looking forward in terms of my perspective on what's important moving next. Number one, I think this notion of continuing to advance evidence and coverage is critically important, both as it pertains to DNA as well as the RNA markers themselves, and both doing this through registries and in a format that allows oncology, both in this country and nationwide, from a molecular testing perspective, to be truly democratized where appropriate. And I say that in large part because one can look at even this week's current events and see that Cigna released a positive coverage decision as it pertains to whole exome. I think that's awfully exciting, and that's certainly what draws attention, but very practically in this country, if one looks at the NCCN guidelines specific to non-small cell lung cancer and can see that version 1, .2016 advocate coverage testing of approximately 7 markers for later stage patients, the simple reality in this country, it's very difficult for labs to receive payment for more than 2 markers, depending on who that payer is. So continuing to do, the basic things well, I think is critically important. And then lastly, trying to better understand as an industry where and when these large panels do have utility in terms of patient treatment, I also think is a very important thing to begin to tackle next year.
All right. Well, great. That was another excellent and very concise set of reflections on the work we've done and what would be useful to work on going forward. We're going to shift to the kind of the Q&A, and because all of our speakers did a great job of staying within the time we asked them to, we have a good amount of time for Q&A, and we have quite a number of questions that have been sent in. In addition to being able to send your questions in the chat box, I think Donna or Jen, I'm not sure if we've actually shared an email address for people who are not on the webinar to send in questions. So it looks like they should send them to Marty Johnson. But of course, if you're not on the webinar, you can't see that email address either. So
okay. If you're not on the webinar and you would like to submit questions, and we do have a lot of questions, and we have a lot of time, so we should be able to get through a lot of these questions, but apologies to anyone if we don't manage to get to your question. But if you're not on the webinar, Marty Johnson, MartyJohnson @cmtpnet.org. You could send questions to her, and we'll get them queued up and hopefully be able to address them. And I'll also just mention, if you want to follow up after the webinar with additional comments or suggestions or asking for more information, please feel free to use that address as well.
Yeah, and also to mention that essentially the Green Park Collaborative is a kind of a voluntary consortium of folks interested in this topic. So essentially anybody who's on the phone who wants to find a way to participate in the next phase of work and contribute whatever insights or perspective, it's fully open to participation. So we'd love to hear from you. We're going to jump right to some of the questions that have been submitted in the chat box, though I did want to give Dane and Mike, since you kind of were the first two speakers and hadn't had the benefit of hearing the other speakers before you made your comments, if there was anything that you wanted, either one of you, Dane, maybe starting with you, is there anything you wanted to kind of respond to that you heard from the later presentations that you wanted to make feedback on?
Well, this is Dane. First of all, I'd like to applaud everyone for staying in what I would consider probably one of the most concise and yet focused presentations of what needs to take place, particularly Mike Kolodziej's presentation on those 6 items. I think those 6 items, Mike, are absolutely should be the point of view of most everyone on the phone call, because I think truly they address the biggest picture in healthcare, which is how are we going to sustain and yet still develop the great and appropriate care of patients. Last thing I'll say is Mency is hopefully addressing many of those things that Mike brought up, and also many of the other things that have been brought up in this conversation, not because we're doing it perfectly, but because we're addressing them at least.
Great. And Mike, any additional thoughts from you?
I really don't. I think I do think, as Dane said, and I would actually echo it. The great thing about this conversation is each of us sort of has our own perspective, and we have our own prejudices. There's just no question about it. But the dialogue has been collegial, collaborative, and problem solving. And if we maintain that, I believe very strongly, and I've been emailing Dane while the other guys were talking, I really think we can figure this out. I really am confident we can do it.
Great. Okay, so let's just jump to the questions, and I'm going to mostly go through them in the order they were received, so partly because I'm just not quick enough to figure out thematic consistency. But we had an initial question about this is probably for Donna, although others on the call may have some knowledge. It's about the CAP proficiency testing, what standards they would use, what metrics would be used to confirm those standards. And then, so Donna, do you want to kind of start, and maybe you can also think if there's anyone that might be on the phone or of the panel who might be able to fill in some details around that.
Yeah, and honestly, in the conversations that we've had with CAP, we haven't gotten to the level. They were still in developing the standards this year, and we haven't gotten to that level of granularity. And so, do we have? I think we do have some folks on the call from CAP. Do we have anybody who can unmute and provide some perspective?
Hi, Donna. This is Helena from the CAP. I don't think that I can add that much more clarity onto the detail of the CAPPC's program. I think we can probably work on or have someone else provide a high-level description of the program. I know that in the process, they are in the process of finalizing it now, so as to date, I don't have any additional details on that.
Yeah, this also, Dane, I know you've been talking quite a bit with CAP as well as the FDA. Is there, and have to think about these standards quite a bit in terms of Med-C. Do you have any reflections on this?
I think that we need to use existing infrastructure to be able to make this successful. And I believe that what CAP is trying to do, and in my conversations with their molecular committee that is working on those standards, I am actually very pleased at where they're going. They're talking about using, shared samples and shared proficiency standards that would, look at a standard tissue a lot like they're doing with CBC proficiency for certain mutations. They are being very cautious in not releasing their initial thoughts until it's gone through a well-vetted process. And so I respect what is taking place internally, but my external discussions, I'm very pleased where we're taking things. I think it would be very difficult for any other group but CAP to be able to do the work that needs to be to really get into labs and be able to verify what they're doing and what they're taking place. What Med-C is starting with is developing high-quality standards, but we're hoping as we develop our standards, which we have been working on in connection with several of the major academic centers, we're hoping that those will mesh beautifully where CAP goes when they release their recommendations sometime in the middle of next year. We feel there's got to be a meshing that takes place.
Right. And
Donna, I'm sorry. Yeah, if the person who asked the question wants more detailed information, certainly feel free to email Marty Johnson, and we will connect you with the folks at CAP who are working on it.
Yeah.
Thanks, Donna.
Donna, also just sort of kind of a related question, and it has to do with, sort of the ongoing dialogue within the FDA developing guidance around LDTs, next-gen sequencing, the IVD, MIA, the in vitro diagnostics guidances. So can you, Donna, at least talk a little bit about how, what discussions took place related to, the role of CAP around proficiency testing, how that relates to, what the FDA guidance and regulation in this space?
Yeah, well, certainly, I mean, I think as everyone on the webinar appreciates, FDA's proposals have been in development. They're being discussed and deliberated, and so while we are optimistic that the final shape of the FDA regulatory framework will be very useful and constructive, at this point, it didn't seem really feasible for us to try to build a set of recommendations around that. But Dave Litwack from the FDA and others have been involved in all of our meetings all year long to provide insight into what their current thinking is, and insight on what a regulatory grade database might look like, or what regulation for LDTs might look like. I wonder if do we have Dave or anybody from FDA on the call who might be able to provide any additional insight right now? I don't see Dave's name in the list. I thought he registered, but maybe he couldn't make it. So, while the FDA has been very involved in these conversations and we've been keeping up with what they've been doing, as I said, we felt that it was premature to try to build concrete recommendations around that right now.
Yeah. And Jeff Allen, is this an issue that you kind of have enough familiarity with? Do you have any thoughts about it? You might still be muted, or maybe it's a bad question for you.
Yeah, no, I don't. I don't really have any specific details, I don't think, that I can really add.
Okay. Well, again, my view is that there's obviously going to be some areas of intersection and overlap between, how what the regulatory framework is going to be around in this space, what CAP is going to have oversight of, and I know from a payer perspective, and Mike, you might be able to weigh in on this, is you don't want to have the responsibility of, kind of understanding, all the nuts and bolts of what's going on there, but you just want to have confidence that the appropriate proficiency and regulatory standards are developed and being met. Is that a fair statement?
Yeah, I think that's absolutely right.
Sean, this is Rich Bender. If I can just comment. I was brought on as a consultant for the FDA area of medical devices in March. And one of the things that struck me actually is the lack of activity that I've seen in this 6 to 8 month period with respect to minimal communications, minimal meetings, etc., and really no dialogue about IVD MIAs, no dialogue to speak of regarding medical devices, chips, next-generation sequencing machines, etc., and actually developing a common standard. I think the work that CAP is doing, I'm a little concerned may actually be done in what may be a possible vacuum with respect to the other regulators. And I'm curious if anyone listening or on the panel is aware that these are being done in a harmonized or in concert way.
This is Vince Miller from Foundation. I don't have specific insights onto that, but it would not surprise me if they were harmonized, because people need to understand that what CAP is talking about in general and what FDA talks about in general as far as standards are just completely different. They're a couple of logs different as far as what's involved. And that is something that I feel like is being, sort of being put under the rug and having a nice conversation on some of these things, but it's a whole different world.
Hi, this is Donna
from
the CAP. I can add to that. Actually, the CAP and the FDA have had several conversations in this space because we want to make sure that the activities that we are doing are not in conflict with what is doing. So we are working hard to try to harmonize and ensure that whatever is developed, at least by the CAP, that it's not going to be overly burdensome. And I also just wanted to clarify here in reference to the activities that what the CAP would be doing versus some of the activities that the FDA may be doing, particularly for the PT testing. That would be an ongoing mechanism to ensure laboratories are maintaining a certain level of performance. So there are other activities that the CAP are doing that will be for initial, are looking into initial validation standards, which is a little bit different than what the proficiency testing would be. So in those aspects, we are having dialogues with the CAP. We actually have been, in essence, when New York State introduced their guidelines, had conversations with them. So there is an attempt in doing so to make sure that we are in conversations with others so that the requirements are either in a complementary form, but they are as much as they can be harmonized.
Yeah, thanks. Well, I think we're going to move on. There's obviously more we need to dig into on this topic, but just there's a bunch more questions, so we'll try to hit on some of those. So there's a couple of questions related to this, the recommendation around, the 5 actionable genes for which there's demonstrated clinical utility being the trigger, to kind of cover the 5 to 50 code. One of the questions was, where did the number 5 come from? What's magic about the number 5? And I think that's, maybe Mike K. or Donna might comment on that.
It's just pragmatic. I think the likelihood of paying for a panel that has 5 to 50 genes, where in that tumor there's only 2 analytes that we meet the grade, that we're not going to do that. I think if there are 9, that's lovely. We'll pay the 5 to 50. I think the point really is until we get some clear kind of uniformity among CMS and all the other payers regarding what the price point is for the 5 to 50, I think that's what people are nervous about, right? They're nervous that, oh gosh, you're just going to backfill, and but the truth of the matter is it gives us opportunity to find a price point where that 5 to 50 panel makes sense for both of us. And so 5 is not a magic number in any way, shape, or form other than it reflects the starting point for the panel based on coding.
Yeah, I agree with that. This is Donna. I don't know what the sort of backstory is in terms of AMA's thinking when they were putting together the code, and I wouldn't call it completely arbitrary, but it's certainly not a magic number. I think it was probably selected because my understanding is that when you get to, depending on the genes you're sequencing, when you get to about 5, that's roughly an equivalence point in terms of the cost equivalency to doing that amount of DNA by older methods, give or take. So I'm assuming that's kind of where the number came from, but it's not a magic number.
I wonder if Vince or Rob, either of you have any additional information on the 5 and the 5 to 50?
I don't. I would just add that, to me, it's not about the number of genes. It's about the other metrics, right? So if you do one of these comprehensive approaches on 40 genes, and I believe this is what, WashU does, they're doing far more sequencing than someone does 200 genes in a hotspot fashion. So to me, it was a needed effort, and it was a first step, but certainly needs some work to really capture the different components here, and a comprehensive approach across 40 genes is so much more than doing hotspot on the whole genome. So there's just some fundamental pieces, but I don't have any particular insights that I can share around the specific question.
Yeah.
Sean, we have a write-in comment from Lee Hilborn who said the cutoff of 5 was that this was an amount where the value of the panel was just more than the sum of the collective single genome testing. So that's basically what I was just trying to articulate.
Okay. I'm going to go on. There were some questions related, and Mike, you sort of touched on this, which goes to sort of like the pricing at which the 5 to 50 would be paid. But it sounds like, Mike, your sort of comment on that suggested that was going to be driven by CMS and not necessarily set by Aetna.
Oh no, not necessarily. But sort of historically, that's the way it's happened, right? I think obviously we're in the midst of a big change because of PAMA and what have you. But no, I think so. It's really interesting, and I am not involved in contracting whatsoever. Thank God for that one. But there's some competition around price, and that makes for an interesting dialogue. I'm aware of that only peripherally. So as I think everybody on this phone call can agree that the cost of the wet lab piece is going to continue to drop, and so I think people like Vince and the folks in his company are going to have to a lot about what that price point makes sense. At what price point does it make sense?
Yeah. Well,
but I don't have any specific insight. No,
right.
I don't.
But just to follow up on that just a little bit, since it ties into, Mike, something you said about, one of your 6 points had to do with sort of the value and, reflecting kind of the value of these diagnostics, not just looking at the effectiveness. And is it your view, that sort of like what's happening on the drug side with sort of move towards value-based pricing, that, that also is something that you're thinking is appropriate, and maybe others in the payer community on the diagnostic side?
I think the potential cost impact on the therapeutic side is so much greater than that is on the diagnostic side. I'm not really that's what's going to happen, Sean. I'm hopeful that when we get that proficiency testing done, the cream will rise to the top. I'm hopeful that we will, for example, answer the question once and for all, what is the best way to do this test? And then I would anticipate, as opposed to a value-based contracting strategy, an all-or-none strategy. That is, people who meet proficiency standard and do the test the right way will be the preferred partner, as opposed to some sort of what could become incredibly complicated negotiations regarding what the appropriate price is for the value brought by any given test. It just gets to be, the degree of complexity just gets to be, I think, unsustainable.
Yep. Okay. There's a number of questions that sort of got to the issue that several people raised about sort of the rapid learning model, which I think Dane mentioned, and data pooling across different initiatives that are looking to gather meaningful data in this kind of NGS-based treatment space. So, and that raises the issue of the opportunities for data sharing, and particularly when they're, organizations that may be competitors as opposed to natural collaborators. So I guess the question here is, do people, what's it going to take, or what are the conditions under which there's going to be a willingness to do, data sharing and data pooling across, different data platforms? And I don't know if maybe Dane, do you want to kind of start with that if you have any thoughts?
Well, I think one of the greatest travesties that's happening in healthcare right now is that we are seeing the great sequestration of data as a commodity, and there's a thinly veiled approach to this that institutions, and many good institutions, are telling their patients, please allow us to use your data so that we can learn from you, and then they don't tell them that the only people that will be able to access their data are through a commercial body. In other words, their data will be commercialized. And so what I see one of the biggest challenges right now is we've got a very much a mind philosophy, to quote, Disney's Nemo movie, Finding Nemo, where everyone is holding on their data and just saying, it's mine, I won't let you have it because I can sell it, it's a commodity. And I believe that is something that people have to get through. Part of the reason why MedC was built as an independent, body was because it truly needed to be independent of all groups so that it could share data. And what the hope is as people see that there is ways of being able to share data, and all of a sudden now a nonprofit organization is having the premier data, sets to look at, and they're open access for people to review, then what the hope will be is that people will stop looking at their own data sets as a large commodity, because they will realize that there is no money in the commoditization, and there will be a greater need to share that data. But as far as I'm aware, we are seeing a great misalignment in the nation. We have different groups that have partnered with different for-profit entities, and each of the groups are just trying to grow their network as large as possible for, quote, research purposes, but also, I will say, not in quotes, monetization, which I believe is a travesty.
Vince, maybe you could talk a little bit more about this. I think you touched on it briefly related to, foundation medicine and just what are the conditions under which it would, make sense for you all to be sort of trying to pool or share data?
Oh, so, we are quite committed to making large bodies of genomic data available. Obviously, on the mixed genotypic and phenotypic data, it requires partners, but I would anticipate you see several significant initiatives from us over the next 3 to 12 months around this, either in specific disease states or broadly. So I think those can only add, and because we don't have time to mine this all ourselves. We know we're missing stuff, but as interested researchers go in there and find certain variants or certain patterns, and maybe they have certain patients where that explains responses that were previously unexplained, that's a win-win. So we're quite open to that.
Yeah. Jeff, at risk of asking you another question that's probably maybe not in your wheelhouse, but I'm just wondering, were there any kind of lessons learned in your work you did on the lung map, initiative, which obviously requires a lot of, kind of pre-competitive collaboration? Any sort of lessons learned there that sort of, facilitate collective effort and, data collection, data pooling, et cetera?
Well, I think we tried to simplify that as much as possible, and I have the good fortune of partnering with Foundation Medicine on the project, so they certainly have the methodologies in place for, collecting the data, reporting it, and, collaborating in the clinical trial space to be able to provide the information rapidly back to the sites in terms of patient stratification, and also have the opportunity to collect the data and biobank the samples for potential future use in conjunction with the Southwest Oncology Group and their sites. So it is a work in progress, but we certainly didn't have to reinvent the wheel given the partnership with Foundation.
Great. Any of the other panelists have any thoughts on this? Because we definitely had, in fact, another question related to this. Someone mentioned the emerging consortia such as GENIE and Orion, and the potential that leads to, competition, fragmentation of all of this genomic and clinical data. And I guess we'll, what I'm hearing so far is that there's some potential reason for optimism that there's a conditions under which these initiatives won't become siloed and independent, but that won't necessarily happen sort of naturally or without some effort to bring them together. Is that fair to say? I guess, Dane, you're really the one that's kind of keeping track of these, I think, most closely.
Well, I think it's a difficult space because what's the incentive for people to build these large networks? And what's the incentive collecting data is not something that is cheap. And so you can't blame groups like Orion who partner with M2Gen to say, we really have got to find a way of being able to do this. And so we've seen a partnering of nonprofit organizations with for-profit organizations. Those for-profit organizations, at the end of the day, have to, show that they are providing value. And so it provides a very interesting environment where, they have to kind of go down that pathway. Now, in a perfect world, there would be money available for the sharing of data across all these initiatives that would allow this to take place. But the problem I see is that, once again, we're seeing kind of a sequestration methodology where everyone's happy to share data as long as you're sharing it with me rather than I'm sharing it with you. And so that is something I think as time goes on, one of two things are going to happen. What is likely going to happen is that we're going to see a few initiatives start to grow and gain momentum, and they become such an important initiative that that people start to look to that as being the primary data source, and anything else will be peripherally to that. Now, whether that's a commercial entity, whether that'll be a private entity, whether that will be something that will come out of the MCI, which has got some pretty amazing things taking place, whether or not that'll be Med-C, time will tell to see that. But what we're hoping is that we don't run into the problem where the data is not available for mining from disparate groups and different groups. You know, we don't want to see data collection initiatives also turned into software companies at the same time. I believe that's a reasonable conflict of interest. You know, the data should be available, and individuals should be able to come in with tools to be able to mine that data, you know, innovate off that data, do the things they need to. And so I think as time will tell, we'll get some better clarity in this whole entire thing. But in the meantime, I don't think there's going to be any, until that happens, we're going to see a lot of disparate efforts.
Yeah. Well, great. I think someone's trying to say something? Yeah. So I think we've come to the end of our time, and I just want to thank all the panelists for fantastic, insights and really helpful comments, and of course your engagement and participation. Thanks to all the folks that dialed in. I think we had a couple hundred people on the call, and everybody did a good job of staying relatively quiet and fantastic questions. We will try to get back to folks with some responses to questions that we didn't get to, because there was a lot of those. And Donna, did you want to make any final comments?
Just want to echo the thanks to everyone. And again, if you want more information about anything you've heard on this call, please do contact Marty Johnson, marty.johnson@cmtpnet.org. Thanks very much to everybody.
Goodbye. Thanks.